Hello, everyone. Thank you for joining us for the March North American Neuromodulation Society Journal Club. For attendees who are listening us for the first time, I would like to say a few words about this journal club and people who are behind of this initiative. The overall goal of the journal club is to bring together neuromodulation experts and experts from other fields with the interest in neuromodulation. We would like to concentrate on three needs and give them opportunity to present their newly published papers. With the perfect attendance and great discussions, I believe this journal clubs already become an excellent platform for building exciting collaborations between basic scientists, biomedical engineers, and clinicians. Our scientific committee is led by Professor Julie Pilitzis, who is a newly appointed dean at the Charles Schmidt College of Medicine in the Florida Atlantic University. Dr. Ilkna Telkis and me, Irina Duff, are co-directors of this journal club. A special thanks to our NANS coordinators, Adriana Zibineller and Kyla Gustafson. It would not be possible to run such a successful webinars without their ongoing support. Before we start, I would like to remind you the few rules for our meeting today. All of us are going to be muted during the presentation. Please write your questions in the question panel on the left side of the screen, and we will be happy to address them in the end of the presentation. The webinar recording will be available at the NANS website in 48 hours after the presentation. I will also have a paper available in the handouts panel on the left side of the screen. Today, we will discuss the recent review paper published by the Professor John Rogers lab at the Northwestern University. We will talk about recent advances in neurotechnologies with the broad potential for neuroscience research. I'm so delighted to introduce the first author of this publication and our presenter today, Dr. Abraham Vasquez Guardado. Dr. Vasquez Guardado is a postdoctoral trainee in the Query Simpson Institute for Bioelectronics at Northwestern University. Dr. Vasquez Guardado received his bachelor in electrical engineering from the Autonomous University of Nayarit in 2007. His master in optics from the National Institute of Astrophysics, Optics, and Electronics in 2012. And his PhD in optics and photonics from the College of Optics and Photonics in the University of Central Florida in 2018. He is recipient of numerous awards. In 2012, he received fellowship from the National Council of Science and Technology to pursue his PhD. And in 2018, he received Dean's Fellowship from the University of Central Florida. He is author of the 38 research publications in high-impact neurobiological and biomedical journals. He currently works in the design and development of bioengineered implantable near-field communication and Bluetooth wireless battery-free devices for biomedical and neuroscience applications. Dr. Vasquez Guardado, we're so happy to have you today. Without any further ado, please take away.
Thank you, Dr. Dove, for your kind introduction. And first of all, I want to thank the organizer for inviting me to this seminar. As you mentioned, so I'm an active member in the Rogers Lab here at Northwestern University. And today I'm going to take the opportunity to share, our recently published paper that came out back in 2020 as a way to collect, technologies that have a great potential for neuroscience research, those that are highly mature in technology development and engineering, but also the ones that have a strong potential for translation in the near future, starting from in vivo animal models, but with potential to applications in humans in the near future, as I mentioned. In order to put my talk into perspective, I will be describing my talk in three sections. So the first one, I will give a brief introduction of neuro interfaces and challenges and opportunities that we find in the field for creating these type of neurotechnologies that uses different type of energy exchange mechanisms, electrical, optical, and chemical primarily. And then I will move with the review of the emergent technologies, which basically is the core of the review article that Irina mentioned a few minutes earlier. And finally, I will describe some of the present and past research activities in the Rogers Lab in the context of electrophysiology, photometry, optogenetics, and photopharmacology, which all of them are current trends in neuromodulation and electrophysiology recordings in the field, which have a strong interest in neuroscience and experiments in animal models. So in order to get this started, I'll give you a brief introduction on what neuromodulation, why. So when we think about neuromodulation, we are describing technologies or methods to interface with the central and peripheral nervous system. So these type of technologies, they look for a way to gain functional access to the brain, the spinal cord, the nerves, and so forth, in a way to understand deeper how the diseases are forming, how to develop a novel type of treatments, or how we understand a pathology or those kind of needs. But also sometimes of these technologies look for a way to actually gain loss of function of sensory inputs, for example. Think about blindness or deafness, which are basically could be genetic or injuries, and as way that we create technologies, we also look for capabilities to gain these sensory inputs, to the body. This type of technologies uses different set of energy exchanges. So traditionally, electrical are the main type of energy exchange for neuromodulation. So this is a traditional way to engage with nerves and cells and neurons, but also chemical. So chemical has been around for such a long time, but we never look at a neuromodulation based on chemical, like a energy exchange. We think about chemical via the use of drugs or pills that can modify some of the neuronal patterns that are in the brain, but not has a technology point of view. So this type of review will also bring some aspects of technology that address chemical in the form of pharmacology delivery to in vivo animal models. Other type of emerging type of neuromodulation uses optical in the form of optogenetics, which uses genetically modified organisms for gaining functional access to neuron cells and circuitry in the brain to understand more behavior diseases and so forth. But all the type of new emerging system for neuromodulation and biosensing uses electrochemical, which actually I will describe later in the last part of my talk. So just putting this into perspective, neuromodulation via electrical stimulation. So electrical stimulation, basically the fundamental mechanism is to change the polarizability or the transmembrane in cells, neurons, and so forth. By doing this, we are changing the way the neurons are going to be firing. We can be potentially inhibiting some behavior or eliciting some behavior or so. In a way that can help us understand more the pathologies or design some diagnostics or therapy tools. This type of neuromodulation have high temporal resolution because of the way a microsecond pulse can be delivered to the brain, with a moderate type of spatial resolution dictated primarily by the density of electrodes that we're going to be creating in these electrical modulation schemes. So the downside of electrical neuromodulation is the cell specificity, which basically the whole cell population that will be within the reach of the potential for these electrodes will be disrupted. So this is going to be one of the downsides that can be alleviated with other type of systems such as a chemical. So chemical stimulation will employ the widely available chemical receptors in the brain, in the body in general. So the delivery and localization, it varies by depending on the pharmacokinetics of drug delivery to those locations. So this is one of the main downsides, because of the drugs that are intended to reach the brain will reach pretty much every other location in the body, unless they are deliberately delivered to that location, via a cannula or so. Has an implantable, like a device or an infusion pump or those kind of engineering aspects. They have high cell specificity, because of the inherent, like a cell receptors in the brain, but they actually downplay with respect to temporal and spatial resolution. And optical technologies are another technique for neuromodulation, and this is a quite example of how like leveraging advances in genetic modifications and synthetic biology can drive the field for creating neurotechnologies. So this technology was completely like unknown before 2005, but after the first demonstration of neuronal modulation via optogenetics on free moving animals was demonstrated in 2007. So the field kind of exploded by creating nanotechnologies that will address this type of need. And primarily the ultimate goal is to deliver light, with millisecond precision to the location of the brain. So that's the ultimate goal. So how we do this one, there are many aspects, many technologies, and primarily optical fibers was the main candidate because that was already mature in the community at that time. This type of approach for neuromodulation have a high temporal resolution because of the millisecond scale that we can deliver optical pulses to the brain, but also have a moderate spatial resolution, which is pretty much limited by the optical propagation in tissue. Diffusion and absorption will severely disrupt the propagation, so it will only limit the stimulation to a very small volume. And on the other hand, we can gain high cell specificity because of this type of genetically modified neurons can be precisely expressed with opsin via genetic approaches. So in a similar way to neuromodulation, there are other aspects to engage with the central peripheral nervous system via sensing. So biosensing in the form of electrophysiology or photometry or electrochemical sensing is another way to know what is going on with neuronal activity in vivo, and which is important to understand how behaviors correlate with neuronal activity, how neuronal circuitries are like a play with respect to understanding a particular disease or dysfunction or so. And creating technologies that can implement this type of biosensing are very important. And most importantly is because the creation of a biosensing plus stimulation will allow us to create novel ways to build or engineer closed loop control of neuronal activity, which are very powerful for controlling, for example, dysfunctions such as like neurological dysfunctions or neurodegenerative diseases and so forth. And just as an example, optical technologies uses genetically modified fluorophores that can, detect ion concentration changes in vivo, as a way to record neuronal activity in that form. But electrochemical type of devices that use aptamer as a functionalization element are very important because they don't not only record electrical activity but also ions, proteins, neuropeptides or so that can be equally important for understanding neuroscience and other type of aspects. So, but when we think about neurotechnologies has a tool that can make it from the lab to the animal applications or human application, we face one of the main limitations, which is biocompatibility. So we know that materials, compatibility is very well proved that is not toxic or so forth. But the challenge is mechanical biocompatibility. So when you think about the brain, the brain is like a gelatin. So you think about gelatin, with as a soft material, and everything that is harder than gelatin will pretty much damage it. So think about putting a metal type of rod on top of gelatin. So definitely will damage it. But if you put another piece of gelatin on top of gelatin, pretty much will not do anything because they have similar type of biocompatibility, mechanical biocompatibility. Same happens when you have very fancy and very advanced nanotechnologies that are made of conventional material. Tungsten microwires, silicon, like a chunks, or even like a soft polymers. All of them have proven, like a performances in vivo applications. No doubt about it. But when we think about chronic implantation, this type of mechanical, biocompatibility or mismatch will severely affect the soft tissues, the brain, for example. So examples of these effects will be in the form of like a creation of scar tissue around the implants. And when we consider electrical interfaces as the way for neuromodulation or stimulation, this type of scar tissue formation will limit considerably the performance of the electrodes. If it is for recording, so it will increase the impedance, so the signal to noise ratio will be completely destroyed. Or if we think about neuromodulation, the charge injection capacity of the electrodes will also be degraded. So at the end, you have a useless type of electrodes, implanted in the body. So opportunities in this field is just creating soft materials. So leveraging polymers, hydrogels, or other type of materials that have comparable mechanical properties than tissue, but also can maintain high degrees of functionalities that can perform the typical type of neuromodulations or biosensing directly from live tissue. So other challenges that we have been observing in the field is how we can increase spatial resolution. So spatial resolution becomes at the need of mapping large areas of the cortex, for example, where there is the need to locate pathological locations in the cortex that will be representative for epilepsy or so. So this type of large-scale mapping of the brain is quite important, but it's very technologically challenging. Not because of the highly packing of electrodes, but the hardware that is needed to run all those electrodes in a free or in in vivo applications and for example in surgeries or so. So another important developments, which faces a lot of challenges in the community, is how we can bring a tabletop type of systems down to a miniaturized headsets that can interface with small animal models for neuroscience research. So take for example microscope or two-photon microscopy or so that that have all the degree of functionalities that their tabletop counterpart, but with a small footprint that can minimally affect the behavior of these animals. And other type of trends that are also coming in Neurotechnologies are the creation of fully implantable, sometimes battery-free devices that can perform different type of neuromodulations or recording in free-moving animals, in free-behaving animals, and the creation of multimodal strategies. Multimodal has the way to incorporate electrical, optical, and sometimes chemical neuromodulations in the same like a form factor that will become very, very important for moving forward different ideas in neuroscience research, molecular biology, and genetics that are emerging in the recent years. So having this small introduction, I will start with my second section of the talk, where I will be describing, like as part of the emerging technologies in electrical interfaces, some of them that will directly answer some of the challenges that I mentioned before, especially in the sense of biocompatibility, a large-scale integration of electrodes, and multimodal integration. So when we are thinking about implantable technologies that will be ideally chronic implantation, so we think about something that will minimally disrupt the natural state of the tissue. And by doing this, so we are looking for materials that have very similar properties than tissue. Take for example soft and tissue-like interfaces. This type of interfaces uses a broad variety of materials. Hydrogels, elastomers, or even conductive polymers. So these type of materials, they share the soft and tissue-like interface, like a capability, but also they have the capability to be microfabricated to include functionalities for neural interfaces. So they can be lithographically patterned, so that they can still engage in a stimulation and recording. So some of these engineering developments maintain low impedances, which is very important and a big limitation in many many type of these flexible and bendable type of interfaces where we need to maintain low impedance, but also in neuromodulation strategies, this type of system also maintain a large charge injection capacity so that the stimulation of the tissue can be the highest efficiency during chronic implantations. The electrical interfaces that now bring, you know, like a huge amount of surface coverage is a great interest, and the idea behind this is that you want to cover a large area with high spatial temporal resolution so that you can resolve neuronal activity, you know, ideally from different locations of the brain at the same time. The way that these type of technologies are or the way that the field is addressing these constraints is by leveraging lithographic and CMOS technologies. So by leveraging the semiconductor industry fabrication methods and bringing novel type of nanomembranes, nanotransistors, and multiplexing approaches in flexible and bendable type of substrates can allow the integration of different or a high density of electrodes that can engage with neuronal activity, you know, with high spatial temporal resolution. Some examples of these type of systems uses nanomembrane transistors, such as 360 and 1,000 even nanotransistors that can record neuronal activity over an area of up to 1 centimeter square. Other type of technologies that uses a CMOS technology for creating electrodes, but also the analog front end for signal processing on the same interfaces is an example of pixels. This type of CMOS technologies, they are like a very minimally invasive, but still rely on silicon electronics to create these systems. Can have the capability to record neuronal activity not only from a granular position in the brain, but also over a broad spectrum of layers, you know, from the cortex all the way to deeper into the brain regions in small animal models, which are the typical models for neuroscience research. Finally, the creation of transparent multimodal interfaces, so that bring the opportunity to incorporate other type of complementary tools for neuromodulation. So sometimes when we are doing neuromodulation, so you care about what is the behavioral effects of the neuromodulation, or if at the cellular level we are observing changes that correlate to these neuromodulations. And examples of these type of devices uses two-photon microscopy or regular fluorescence microscopy, optogenetic stimulation, or even hemodynamics recording. At the same time that patterns of neuronal activity are being recorded by these type of electrodes. So the transparency of these electrodes is basically a requirement, and at the same time enables the use of the other multimodal approaches. So these type of examples can be easily deployed in free-moving animals or head restrained animals, where these complementary tools can be used hand by hand to understand even better the behavior or the physiology of the animal or the experiment under question. So with this introduction of electrical interfaces, so I will continue with the optical interfaces. We basically received the call for optogenetics. So optogenetics, as I mentioned before, requires the precise delivery of light to the location of interest in the brain, and having the method to guide light to those locations is very important for understanding neuronal circuitry and behavior, and correlate with behavior and so forth. Optical fibers are very powerful to do this, but they have many limitations that I will not mention here. But one of them is that there is only one excitation port, which is at the end tip of the fiber. A different type of novel technologies for delivering light from external sources to the brain uses tapered fiber, which basically is like a cone that gradually shrinks to almost a few microns, at the tip. At different locations of these tapers, so there are many exit ports that can be individually addressed via excitation of that fiber, and you can gain, like a multiplexing or delivery of programmable multivision ports for stimulating neurons or brain at different depths, for example. Other aspects for achieving this is the use of optical waveguides. So optical waveguides uses silicon photonics or other type of microfabricated systems for achieving the same purpose: deliver light at different locations in the brain, and with high, like a precision and so forth. So, but another aspect for delivering light is not coupling from the outside to the brain, but is to deliver the light source directly to the tissue or the brain. And this is what we have been leveraging in the lab for the past a few years, is the use of microscale inorganic light emitting diodes. So these micro LEDs can be as small as few tens of micrometers in the footprint, but can still deliver can drive neuronal modulation that can be observed in the modulation of behavior, as we can see in the heat maps for behavioral experiments in optogenetics. These type of systems can grow, or these type of light sources can be grown in monolithically in silicon wafers, and we go to where we will face some issues with biocompatibility, as I mentioned before. But other type of integration includes flexible probes. So this type of flexible probes can host the micro LEDs, and sometimes additional type of interfacing. So in the form of temperature sensors, electrodes for electrophysiology recording, or even photodiodes for photometry in the same probe that allows to create like a multifunctional type of system for a recording and stimulating neuronal activity. These type of micro LEDs become very, very powerful when we couple to wireless power transfer and wireless communication mechanisms that are now at the turning point for delivering neurotechnologies to neuroscience research in vivo animal models. These type of devices uses in inductive in magnetic inductive coupling, to harvest power from an external source to an implantable device. And as a result, the device is totally subdermal and battery free. And by managing the amount of power that you are harvesting, you can implement different type of integration. Some of them for stimulation via optogenetics, or sensing via photometry, or even has a closed loop control of physiological condition via like a multimodal type of devices. Most importantly is that these type of devices are highly miniaturized, that can be used in many type of animal models, from mouse, rats, birds, or even fish have been demonstrated in the past. And most importantly, it goes to not only to the brain, but also to peripheral nerves, the peripheral nervous system, or even peripheral nerves. So, such as in the example of one implantable device that is stimulating the spinal cord of a mouse, and modulating nociceptive behavior on this paradigm. As I briefly described earlier, so the need for creating highly integrated systems. So it's another important aspect of neuromodulations, not as the form of implantable probes or implantable devices, but has a miniaturized system that can perform 2D and 3D imaging in vivo and free moving animals. And examples of these ones, of these type of technologies, uses wide field microscopy that leverages a calcium photometry, for example, or even two photon microscopy that uses advances in electro modulators, for example, that are able to bring this light, high energy pulses, down to the brain, and perform this type of imaging, with high resolution and high temporal precision. Most importantly, there are other type of device approaches that they don't fall under the category of miniaturized devices, but they uses the capability for holographically pattern an array of stimulating pores in vivo animal models for precisely stimulating neurons at the same time of recording from the same type of animal. So this type of holographic approach will be a quite cool interesting technology in the near future. Hopefully, somebody in the field will be already working on this. So my next section will cover multimodal interfaces, because in most of these cases, multimodal interfaces become a supportive technology within each other. Some of these examples are the use of polymer fibers. This type of highly functional polymer fibers can incorporate different neuromodulations and recording strategies. So this, for example, on the left uses light delivery mechanisms via optical waveguides, but also it uses microfluidic channels for delivering fluids, drugs directly to the location of interest in the brain. And at the same time, it uses electrodes for electrophysiology. So these type of systems are important in the sense that, you know, there are type of behavioral paradigms that are under hypotheses that require the validation of this type of the hypothesis via two different mechanisms, typically inhibition and excitation. And this type of approaches can be easily implemented in this type of multimodal type of interfaces. Other aspects of photopharmacology type of delivery is the use of wireless power transfer and highly miniaturized devices, as the one that we have worked in the lab in the past few years. This type of devices uses a filament optofluidic device that can deliver up to 4 different drugs to the brain. And these drugs can be programmable at different, you know, like a flow rates and temporal delivery, so that, you know, we can study more in depth, you know, what is the effect that they're having in the behavior of the animal. But that's just one example. But eventually, these type of systems are looking for technology that can be eventually translated to humans in the near future. Take, for example, the device that we have on the right, where the opioid receptor agonist DA-NGO is delivered to the VTA, and delivering this fluid will elicit motor rotation on the animal, which is observed in the heat map in the device, which is a behavior that is not observed in the control experiment. So the final part of my review will bring together different aspects of biosensing, more than just electrophysiology using electrical interfaces. Has the field of synthetic biology and genetics you know, advances, new proofs for biosensing are appearing. For example, the use of photometry is one of these examples, where the field or the community uses genetically modified fluorescent proteins. These fluorescent proteins can be expressed with high precision in the membrane of cells, neurons, or other type of targets with high precision, and their fluorescence is modulated. Fluorescence efficiency is modulated by the capture of the ion, the target ion. Examples are calcium, you know, like indicators, but more recent proofs are in the context of neurotransmitters, dopamine, and so forth. But at the end, the mechanism, underlying mechanism, remains the same. Has the probe is capturing this type of ions, you know, in the surrounding of the fluorophore, the intensity will change, and that's the way that we can detect in real time or monitor in real time the pattern, the activity of the neurons, so we can see in the graph on the right, in the top right. The other examples of sensing uses electrochemical, and this type of electrochemical, you know, technologically leverages the development for electrical interfaces, no problem about it. But they are strongly you know, enhanced by the use of aptamer functionalization. This is one example. And the main advantages of aptamers is that they can be synthesized, artificially synthesized, to bind almost anything. So it could be ions, proteins, peptides, neurotransmitters. You know, you can mention, you know, like hundreds of them. So the main you know, like a goal of this type of technology is to bring functionalized electrodes or sensors directly to the brain or other part of the body, or even, you know, collect physiological fluids and do the assay there. And by doing that, we can actually be monitoring neuronal activity indirectly, not has the electrical potential, you know, recording, but has a way that the brain is releasing different biomarkers in the form of chemicals or so. So having this short review, I will proceed with my next section, which is you know, recent advances or works that we have done in the Rogers lab in the past few years. And some of the specific examples that I will be describing here is the use of a large-scale microelectrocorticography for non-human primates, you know, in vivo studies. a wireless photometry directly from mouse, in a free moving and free moving paradigms, as well as a smart near field communication implants that can enable social studies in group of mouse. And finally, I will give you a small example of a programmable for pharmacology, which is a recent work that the lab has created, is currently under revision. So as I mentioned, earlier in the talk, so there is the need to increase the coverage, surface coverage of the cortex of the brain for many reasons. So one could be just to understand how neuronal dynamics are happening when the subject is behaving or is being stimulated, but also has a way to interrogate neuronal activity that will report like a nodes or focal points for epilepsy, for example, which is one of the greatest tools for surgery, tumor resection surgeries, for example. So regardless of the interest of the field has been moving forward to creating technologies for different type of applications, but the main underlying engineering challenges is how we can create a large array of devices with high precision and high functionality. So the lab was involved in this project back in 2020, probably earlier than that, where the system leverages nanomembrane transistors. These nanomembrane transistors are typically in the order of few tens of nanometers, no more than 500 nanometers. These transistors can have different functionalities in the operation of the device. One is the use for recording the physiological signal. The other one could be for implementing multiplexing. So instead of having 1,008 individual lines for addressing one individual electrode, so the matrix is the device is configuring a matrix so that we can reduce the amount of connection ports to the device, which actually decreases the burden of the hardware that is needed for driving these systems. The devices, these type of devices are mounted in flexible substrate, which is another big, important aspect for biointegration. And this type of flexible interfaces can allow to conformally code the highly irregular topology of the cortex, for example, while at the same time recorded with high fidelity and high, precision and neuronal activity in vivo. For example, we can have here a video where this device was recording neuronal activity in vivo from a non-human primate while doing visual stimulation. So how we can see here in the video, the 1,008 individual electrodes are collectively recording neuronal activity that will be representative for this behavioral like a study. But at the same time, this type of device could be, could be showing, how the focus of epilepsy could be happening in a model as well. So this device covers an area of roughly a centimeter by centimeter, square, and the technology promises a scalable, like a increase to a few thousand of these electrodes for a large, like a high density recording of neuronal activity. But most importantly, the driving part of this technology development is the need for neuroscience research. A lot of the work that we do in the lab is basically pushing the limits of engineering. But most of these engineering developments have a strong impact to biology, and that's when our collaboration with neuroscientists, with doctors, and so will implement a nice synergistics for creating these type of devices for addressing big constraints in biointegration, in capability, sensitivity, and integration for uses in vivo. Other example that I've been involved, this one I've been involved personally in the past, is the creation of implantable wireless photometers. So photometry, as we know, it uses fluorescence as the mechanism for biosensing. Changes in fluorescence will correlate to changes in the calcium concentrations in the vicinity of the probes, and these changes in calcium concentration are basically indicative of neuronal activity. And creating a implantable probe that have that leverages the developments in micro scale inorganic LEDs, micro LEDs and photodetectors can easily implement this type of technique in vivo. The devices of this form have an implantable needle that contains a micro LED for stimulating the neuropopulation, around the device, and induce for fluorescence. An optical filter will and in conjunction with a photodetector will get the signal coming from the green fluorescence, and the photocurrent will be transmitted to a microcontroller or a microprocessor. These type of devices can be formed in a totally battery-free and fully implantable form that can be deployed directly to free-moving animals, such as the one we see in the picture. And most importantly, these type of technologies have similar performances than state-of-the-art fiber optics photometry, which is the typical technique for photometry in neuroscience. And at the same time, these type of devices can be recording data in real time and during even extreme experimental paradigm for validation. Another example that I was involved personally in the past few years is the development of a smart NFC device for social studies. The use of wireless and implantable devices for optogenetics has been, in the field for quite a few years now, but there was a main limitation of these type of devices. There was no the need for individual addressing of a population of animals. So these type of devices, they leverage wireless power transfer, and when when the animals are enclosed in a particular experimental enclosure, the devices will all at the same time will be activated with no distinction whatsoever from which device is of interest. The development that I did in the past few years was to create an implantable device with a microcontroller and NFC communication capability that can individually address every individual in the experimental enclosure, and at the same time we can address a programmable illumination protocol that can be changed in real time, on demand with the use of a computer interface, so that we can test a large cohort of animals in the same, sharing the same experimental enclosure. And this way allows to study more interesting phenomenon in neuroscience, which is in the context of brain synchrony. So it was hypothesized that like stimulating the middle prefrontal cortex with the same pattern on two different animals will drive a certain degree of brain synchrony that will be observed as increases bouts of social interactions. So we tested this hypothesis using the devices that I developed in a pair of mouse, and we observed that during during an episode of synchronized stimulation, meaning both animals are receiving the same stimulation pattern, spend considerable amount of time doing social interactions. However, when this synchrony is disrupted on the same group of animals without actually having to change another group, the amount of time that they spend doing social interaction considerably decreases. So this difference is summarized in the graph that we can see in the bottom part, where the red shades colors represent any type of the categorized social interactions that can be observed in mouse, while the green and blue shades correspond non-social interactions. White spaces just correspond locomotion in general. So when we compare the results from social interaction during synchronized stimulation, almost 50% of the time the animals spend doing social interaction, in comparison with 45% when these animals are not, synchronized. And this type of desynchronized behavior pretty much correlates to a control group where there is no optogenetics, active in the same location of the brain, which is basically very interesting technology for moving forward, like these frontiers research paradigms in neuroscience. Just to move a little bit forward, my next example is the use of a multimodal optofluidic system for uncaging glutamate. So we know that recent developments in neurobiology and synthetic biology uses photoactivatable drugs, which are totally inert when they are in the brain, for example. But only by the induction of photolysis, the cage and the active element can dissociate, and the active element can become active for neuromodulation. For example, ruby glutamate, which is totally inactive, when it is in the cage state. When it's delivered to the tissue, to the brain, it can actually release via photolysis and become free glutamate that can simulate artificial glutamate release in the neurons, and because it is a neurotransmitter, can actually induce modulation of behavior when delivered to the location of interest. So in order to test these type of devices, we develop a system where it uses microfluidic systems for delivering this type of photopharmacology compounds, and in combination with light stimulations. So by delivering this type of compound directly to the motor cortex of animal, and followed by the stimulation of optical stimulation, we can photolyze glutamate and eventually become it active, so that it can induce modulation of behavior, as we can see in the graph on the right. But delivering just the photoactivatable compound without any stimulation, or the other control experiment, will not elicit this type of modulation. So basically, leveraging the technology for light delivery and drug delivery in the brain can give us an additional degree of freedom for neuromodulation that otherwise will be quite impossible, because of the pharmacokinetics, transport of drugs to the location of interest, and as well as absorption and scattering of light propagation in tissue, which basically transcranial stimulation will be off the chart in this case. Finally, I will conclude with the next exciting experiment that was published a few years ago in the Rogers lab, also in the context of closed loop control of physiological activity. In this case, a fully implantable wireless powered transfer device can engage with bladder control to implement a closed loop system for controlling aspects of urinary inconsistency or overactive bladder activity. So the device of this sort includes a sensor in the form of a gauge, strain gauge sensor, which is basically a resistor that varies with respect to the contraction or expansion that it can experience during the physiological event of the bladder, and an optogenetic stimulator that will be able to counteract this type of system. The device can detect voiding events, as we can see in the graph on the bottom, and based on the periodicity of these voiding events, the device will be able to engage in a closed loop and actually counteract this activity. The examples that we can see here is a behavioral experiment where a pair of animals, one negative control, the other one positive control, is delivered with cyclophosphamide. So this compound will induce like avoiding dysfunction, and has the voiding events are happening, as we can see in the vertical lines in both populations. The closed loop control will kick in and will try to counteract it, which we can see in the positive control graph in the bottom side, but not in the top side. So the main thing about or the main message of this type of closed loop control is that we can create technologies that can interface with the biology at different degrees of complexity, and doing this type of integration, we can address different like questions in pathologies, in dysfunctions, and so forth, that will bring these type of technologies to a next level of impact to the society. So with this, I will conclude my talk. So the main take-home message that I would like you to have from here is that the next generation of neural interfaces will pretty much benefit from novel materials, novel composites, novel type of like a soft material that will drive the engineering for creating these type of systems. Another important aspect is that like a multimodal system. So some of them are supporting to each other, are emergent research in neurobiology. So sometimes we don't think about electrophysiology by itself. We can think about electrophysiology plus optogenetics, or calcium, imaging, or so forth. So this type of redundant type of neuromodulation and biosensing are important to actually make a better impact in the paradigms that neuroscientists are studying in vivo. And finally, the emerging neurotechnologies, have the potential to not only to revolutionize the way we study, neuroscience, but also to revolutionize, the way we diagnose diseases, pathologies, dysfunctions, neurological, diseases, and can drive the development of novel treatments and interventions in surgery. And we have been observing this one in the past few years in the commercialization of different type of technologies, for example, deep brain stimulators or spinal nerve stimulators and so forth that definitely are making a great impact to the society. Thank you, everybody for for your attention, and also I want to acknowledge my lab, my set of collaborators that made this type of work possible, and grad students and other grad students that actually contributed indirectly to this talk. So thank you.
Thank you so much, Abraham, for such a fantastic presentation, such incredible inventions. So while we're waiting for questions, we have 6 minutes for questions. Let me ask you some of mine. Can you go to your arrays slide? Your large-scale ECOG arrays? Just few, yeah, 5 slides, 6 slides before.
Slides number.
I don't know.
8?
I guess. Yeah. 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 Next one. Yeah. So I'm just curious. Asking engineer, are they the same? Always was curious about that. Are they the same size? Like, are they made it the same size? You said 1 by 1, or you can make them custom size.
Well, yeah, for the engineering point of view, so the surface configuration can be arbitrary. So has just an initial demonstration and to demonstrate the capabilities. So they are typically done in a square array, but in reality, you can actually pattern the electrodes. So you can have, for example, higher density in one section where you know that there will be more neuronal activity, and you can put them in a more dispersed, like a configuration in other regions where you know that you don't care about too much the intensity. So basically, the patterns are arbitrary because at the end, this technology uses microfabrication and lithography. So you can create your fabrication, strategy to accommodate the final, distribution of electrode that you want.
So the distribution of the electrode doesn't depend on how it's going to record. So it's going to record anyway. So what you just said, we can either make a different size or make a density in certain areas more or less. Yeah. So how about so can we make like I guess if we, for example, want to target the certain area, just for example, somatosensory cortex, nothing else. So we're gonna to activate electrodes which are gonna to touch that or be on the surface of the somatosensory, or we're gonna to just have the size to cover. So like how to make it specific to certain area.
Okay. So if I understood correctly the question, so how you can design the electrode array that can precisely record neuronal activity for the desired location of the brain.
Area of the brain, like motor cortex or somatosensory cortex, is it possible?
Yeah. So well, I mean, this one comes at the expense of like a precise placing of the electrode, because that the electrode is just like an array, and wherever you place it is the activity that it will be recording. So you can guide the engineering of the electrode so that it would be easy for you to locating the area of interest. But the device at the end will just record whatever is in contact with.
Yeah, this is the thing, the question which discussed frequently in engineering field, that with any tungsten or tetroids design old-fashioned. So it was, like you said, it was specific. You just going into that area, but it doesn't cover the big surface. With this arrays, you can go put them on the surface, but it's like it's covering the big area. So just an idea to think about. There are a lot of other questions. For example, can you incorporate optogenetic into this arrays, or it's something absolutely out of fantasy? Like if it's going to be just purely electric recording, or actually it's possible technically to incorporate, if we're covering big area and we want a certain area to be stimulated, like certain portions of this, let's say, motor cortex to be stimulated, another not.
Well, I think that it could be possible, but probably not with this platform, because the platform or the device is somehow opaque, so light will not propagate directly through the device. Sometimes optical stimulation will also elicit some like artifacts in the recording. So the high density will pretty much affect this recording while doing simultaneous stimulation. But let's say that you sacrifice density or resolution, but and you can replace certain locations of this array with micro LEDs. So now you can have, for example, an array that has probably 8 by 8 or 10 by 10 micro LEDs distributed across the pattern, and you can deliver the stimulation on those micro LEDs while you can maintain recording in the other. So that could be possible. Another possibility is to change the type of engineering for the material, and you can for example replace for optical like optically transparent electrode and materials that can allow as I mentioned before to detect light propagation through the electrode. That's another alternative, but that would require probably like a light stimulation via projection or so. So yeah, so it is possible, but so most of the time this type of question will drive an engineering effort, and basically a lot of the engineering is driven by the requirement on the neuroscience counterparts.
Exactly, because we either do precise manipulation stimulation with optogenetics, where we don't need all this high density arrays, or we're doing that just electrophysiological mapping, which this array is for. So just thinking, the next thing could be combine them, because sometimes if you mapping, you can actually manipulate. So it will open a completely new line of research, especially for people who are working with the cortex, or with superficial area.
That's true.
I guess I'm so interested in that. Can we make them wireless so that these arrays are tethered? Is it correct, that particular design? Are they wireless?
So this type of device is a wired, because one of the main challenges in the field for electrocorticography is the density, and when we think about density, we're thinking about individual individually addressable elements, and for each individually addressable element you need an analog front end material component for digitalizing amplifying and filtering and digitalizing the data. So making a totally wireless, it could be possible, but sometimes you cannot implement them in a battery free format. You can do it in a battery operated format, and you are envisioning like a kind of like a bulky type of headset that can incorporate the communication, wireless communication with all the signal processing and other things. So in that cases, yeah, it could be quite doable, which is very tolerable for non-human primates, for example.
And one last question for this great technology. Does it exist for spinal cord, or we can do just use this in the cortex? Can we do the same thing for the spinal cord?
So technologically, this device can be adjusted to the application. So there is no, at least from the research that have been done, so there is no a device that have the form factor for the spinal cord. But technologically, like speaking, so the translation for a ECOG to a spinal implant could be quite straightforward. So it's just a matter of rearranging again the distribution of electrodes, probably working a little bit better with the mechanical compatibility, because the spinal cord is a lot more challenging than just the brain, and just leveraging the same like a mechanism for like a interfacing, and that should be quite doable.
Yeah, because spinal cord has also quite complex circuits, and people don't pay as much attention to them. It's interesting. It would be interesting to map. It's less complex than in brain, but it's also interesting to study. Oh, we're 4 minutes over time. I do have many other questions, so I guess I will get a chance to answer to ask them in another time. So I would like to conclude our webinar today. Thank you so much for coming, Abraham. We are very excited to have you today. Thank you for everyone who joining us today, and stay to the end of the presentation. And bye, everyone. Enjoy the rest of your day. Bye-bye.
Okay.